everyone. I am Dr. M.Parimela Celia, Assistant Professor, Department of Microbiology, St. Joseph's College of Arts and Science, Autonomous Cuddalore, Tamil Nadu. I am going to present a topic on laboratory hazards. Now let's see the introduction. The infection of laboratory workers is a common problem while handling the microbes. The problem is found to be more severe in case of this clinical microbiology labs because those workers used to handle more pathogenic cultures. That means they may induce pathogenicity, that is, they cause disease. And common lab infection causing microbes are Brucella abortus, Brucella melitensis, Pasturella toleransis, and Salmonella. And the top lab occurred infections are said to be brucellosis, Q fever, hepatitis, typhoid fever, tuberculosis, dermatomycosis, psittacosis, and coccidiomycosis. And all these infections are occurred due to inhalation of aerosol that is generated during the work. And now we'll see the three possible routes of lab acquired infection. The first and foremost thing is the inoculation, direct inoculation into the host, that is through needle injury or incision or break with a sharp instrument or with broken glass. Second one, ingestion, that is intake. This may happen due to accidental swallowing of infective materials, that is, if they perform mouth pipetting they will be at risk. Then the third one is inhalation, that is through breathing the dust particles or aerosol containing pathogens are inhaled, naturally they will get respiratory infection. And now we will see the hazardous procedures. First one, the withdrawal of needles from vein. This may happen while collecting blood samples. And next one, the culture may liberate aerosol on vibration. So this may happen while opening a test tube or conical flask containing cotton plug. So naturally the aerosol will be generated on vibration. Third one, the content of syringe is forcefully ejected. After collecting the sample, blood sample, and if it is ejected forcefully, naturally they create aerosol. That is a risky procedure. Then if last drop from the pipette is blown out, so blowing out the pipette is a wrong practice. So they may disseminate the pathogen into the surrounding. Then spillage of cultures, so this will happen by mistake. So if accidentally, if there is the breakage of test tubes or conical flask containing the culture fluids, naturally the infection will spread. And during centrifugation of culture fluids, they also generate the aerosol due to the vibration, because the culture fluids undergo vibration that may leads to the development of aerosol. Then freeze drying, this is culture preservation method, so which may also generate aerosol. Then opening of sealed ampules, that is opening of cultures, that if you brought the cultures from institutions and if used to open the new cultures, either MCC or MTCC, by opening the ampules, naturally there is the development of aerosol is possible. Then removal of cotton plugs which is wet. So this is a very wrong practice. Naturally we should not allow the cotton plug to get wet while sterilization. Otherwise surely they will be producing aerosol while opening the cotton plug. Next, accidental dropping of culture plates, tubes. So this is another thing that may happen by mistake or by unknowingly. If we spill the culture, naturally this may leads to a severe spread, direct spread into the environment. And now we will see the hazardous groups. The lab acquired infections are classified into different groups based on the risk of infection posed by them. The five major agencies, they have given the classification. First one, United States Public Health Service. Second one, World Health Organization, WHO. Third one, United Kingdom Advisory Committee on Dangerous Pathogens, ACDP. Fourth one, European Union, EU. And the fifth one, European Federation of Biotechnology, EFB. So all these agencies except this EFB, they classified the microbes into four major groups. Now we will see what are those. First one, group 1, an organism which is most unlikely to cause human disease. That means naturally they won't cause any infection to human being. And the group 2, an organism which may cause human disease but unlikely to spread. That means they cause the disease but they won't spread. And effective prophylaxis, that is the preventive measures and treatment are available. Then in group 3, an organism which may cause serious human disease and they may spread in the community. They cause the disease and also they spread in the community, and there is the effective prophylaxis. Preventive measures are there and also the treatment is available. So this comes the group 3. And the group 4, an organism that cause severe human disease and may have high risk of spread in the community. That means they will cause the disease, also they spread in the community, but no effective prophylaxis or treatment is available. So this is a dangerous one. And now we will see the classification proposed by EFB, European Federation of Biotechnology. And now we will see the EFB group 1. In this, the organism that never been identified as a disease causing agent for human. So that means they won't cause disease. It is not the pathogenic one. Then EFB group 2, it is considered as low risk organism. That means they cause disease to man, especially the person who is working in the lab. They are unlikely to spread in the environment and prophylaxis and treatment are available. Then the EFB group 3, it is the medium risk organisms. This one causes severe threat to the lab workers, and the prophylaxis, that is the preventive measures and treatment are also available for this group. And the EFB group 4, they are considered as high risk microorganisms. They cause serious illness to lab workers and spread to people. In this effective treatment and preventive measures, it is not available. Then group E denotes the environment. That means it is giving more severe threat to environment than man. Mainly this leads to economic loss. And now we will see the prevention of lab hazards. Lab hazard can be easily prevented if we use the biological safety cabinets. It is classified into 3 types, class 1, 2 and 3. First we shall see the cabinet class 1. This allows the unsterilized air to pass into the cabinet and also around the materials within, and it sterilizes only the air to be exhausted. That means it will purify the air which will be released from the safety cabinet. Then the cabinet 2, it is in this the air in the room is filtered and sent into the cabinet. That means outside air is purified and it will be sent inside. Then in the group 3 cabinet, air coming in and going out of the cabinet is sterilized by using the special filter, HEPA filter, high efficiency particulate air filter. So this will handle that is the infectious materials are handled in this cabinet with the rubber gloves and it is attached and sealed. So it is we can easily insert our hands and we can walk. So this is a good one and it is very effective in the prevention of the spreading of infection. Now we will see the hazards in industries. During normal industrial fermentation, the microbes may escape into the environment, mainly in the form of aerosol. So this is more prone in the downstream area, that is the product recovery area. And also the contamination chances are more in the airspace, that is at the top of the fermenter. And also more spreading is possible, that is leakage, accidental breakage, they may occur in the fermentation vessel. And also the amount of aerosol produced depends upon various factors such as the stage of the fermentation, that is the initial stage or the middle or final stage, also the size of leakage. So how we can control this? So the control measures can be carried out by 3 ways. First one, physical containment, that is we can use the closed rooms, also filtering the air from the working site, as well as sterilizing the contaminated material, so that will reduce the escape of microbes. And second one is the biological containment. In this, the risk is reduced by using microbes that cannot grow outside the culture medium. So we have to select the culture in such a way so the risk can be reduced. Third one, by using the safe techniques including lab workers for training. We have to train them so they can do the work very carefully by handling for that is especially handling of cultures as well as handling of equipments for analytical purpose. And also the drop or jet of fluid should not be allowed to fall down from the pipettes. So this is a important one that can be prevented. Used pipettes and slides should be discarded only in the disinfectant. Then fumigation of the rooms, it has to be strictly followed. And also disinfection with 70% alcohol or hypochlorite can prevent the formation of infectious dust. And we have to check the tubes that will be used for centrifugation for cracks. If there is any crack, you should not use that. And also we can use the filters, HEPA filters, either single or double, and that is should be provided for exhaust system in order to prevent the release of microbes in the environment. Then appropriate vaccination has to be done in order to reduce the risk of infection to make the workers safe. Thank you.